All right. Okay. So let me open up the slides too. And also I may need to show you some protein structures and some links as well. So let me open up this one as well. All right. So last time, if you remember, we talked about the general idea of protein structures. We have looked at inside of some protein data bank PDB files, and we have seen that protein structures are really, it gives us atomic coordinates of amino acids that make up these macromolecular structures with tens of thousands of atoms. And the protein structure universe is so diverse that there are also very small proteins with 30 amino acids, 40, 50 amino acids. There are very short proteins. There are proteins on the average like maybe 300 amino acids. There are proteins, 2,000, 3,000 amino acids, not a single chain maybe, but we are going to see this hierarchy of protein structures, starting with the amino acid sequence, how the analysis to the functioning, how we are going to see a functioning protein structure is composed of modules, which are called subunits, which are individual amino acid chains themselves. They form this quaternary structure, which we call the fourth level structure, is the composition of these amino acid chains, subunits, into this larger functioning machine. That largest functioning machine is the quaternary structure at the top level. Then if you look at this individual amino acid chain, one of those subunits in that macromolecular structure, it has a three-dimensional shape, and it may also be very unique. And that's the tertiary, the third level structure, which is called the tertiary structure. And if you look more inside that tertiary structure, you're going to see those local shapes that we have seen last time when we looked at protein structures, the helices, the parallel beta strands on top of each other as a sequence of sheets. So those beta strands, alpha helices, they are the secondary structure, second level. As we go down from the fourth level is the combination of those formation of the functioning protein structure, but possibly with multiple protein chains. There are also single chain protein structures, which means that it's fourth level and third level is basically the same thing. Because if it's just a functioning protein structure, but it has a single amino acid chain, then it's also it's fourth level and third level are the same thing. But there are also protein structures, as I told you, which is composed of a combination of just multiple things. You probably are too young to remember that cartoon movie, but when I talk about these protein structures, I remember in my childhood we had this animation, the Voltron. Uh, Voltron was composed of multiple robots becoming a large humanoid robot. These small robots were like just lions. They're also robots, but they had these lion shapes. There are 5 or 6 of them. So one of them makes the head, the two are like arms, the torso. So this is the protein structures are just like those. So the functioning thing is the Voltrons. The Voltrons, the quaternary structure. Think about the subunits are those lions. They also have like individual function, they have individual shapes. They are separate and then come together. So those lines are like the third level tertiary, and they have also a three-dimensional shape. But if you look at the inside, there are also some local shapes that you see. All of them has a leg, for example. Those legs that you see in all of them, they're those local structures. How those legs are oriented with respect to each other, we may not know that. So if you just know that it has legs and arms and this, you may not know immediately its three-dimensional position, how it's positioned. But yeah, that's the same thing with secondary structures. Secondary structure information is just actually a one-dimensional information saying that these amino acids are part of a helix. These amino acids are part of a beta strand. These amino acids are this. So that's again like sequence information. It's one dimensional. Although its name is secondary structure, the information that it provides is just that this amino acid is part of an helix. This amino acid is part of a beta strand. So though it just tells us that which local shape that it belongs to. How those local shapes, how three helices, how they're oriented with respect to each other is a three-dimensional information. So from that 1D information, we jump to three-dimensional information in tertiary structure. So we are going to, the slides are all about, the first part of the slides are all about the details of protein structures. There are some chemistry details, how peptide bonds are formed, what kind of dihedral angles we have in a so the chemical bonds, their lengths are fixed, but their the angles that they make when these carbon nitrogen it makes bonds with its side chain, the angles may change. So all these things are the chemistry details. You don't need to know all the details, but I'll just go over them quickly because knowing that these are really the protein structure is nothing but really a chemical structure. It's a compound, but a macromolecular, a very large compound, and knowing how it's formed, what kind of chemical bonds it has, may be important if you're going to study protein structures later. By the way, if you have any questions at any time, please feel free to write in the chat box. And I'm going to, after we finish protein structures, I will give you a programming assignment, which will not be very difficult. It will just be processing one or two PDB files. I'm given a PDB file. You'll write a generic program which will work on any PDB files, any PDB file, and it will read those atom coordinates, pick up certain coordinates of specific atoms that I will specify in the assignment, and do some analyses on that three-dimensional structure. For example, computing the width and height of a protein structure. Which means that how do you compute the width? What does width and height mean? We should first define, but maybe I can may simplify it. Compute the diameter of a protein structure, which is find the distance, the longest distance between any two pairs of carbon alpha atoms. That may be something like that. Okay, so it will just be an analysis type of protein, this analysis of a three-dimensional structure in terms of its geometry. I'm not going to give you anything about physiochemical properties or some electrostatic potential. You're not going to be doing that kind of an analysis, but you're going to be doing some geometric analysis on the protein structure provided as a PDB file. And protein structures are really like three-dimensional shapes, really, ignoring their chemical properties, of course, but we cannot ignore always the chemical properties, but for simplicity in this assignment, we're going to do that. All right, so that's also a heads up on what we're going to be getting as a programming assignment. So protein structures, we have seen last time, I'm going to skip this motivation part. We have seen lots of examples how they are like macromolecular machines that govern the most of cells function. They act like enzymes, they act like, they form the structure, they are like ports, communication portals with the extracellular, intracellular, so outside the cell and inside the cell. So the lipid membrane contains these pores. We have always learned in high school that, for example, the animal cell or the cell membrane is semi-permeable. What it means is that some things may pass, some things may not. And how that semi-permeable property came about, we never discussed in high school. But here we see that what makes it semi-permeable is there are really like portals on the lipid membrane, which are made up of these channel proteins. There are proteins that are called channel proteins. So they are situated in the membrane. They have circular shapes, and I know one such protein, which I actually visualize in my, it was one of the large proteins that I visualized during my PhD. So this one, or was it zero? Yes, this one. So I remember its shape. So it's like uh, a conical shape. It's like not entirely cylindrical. So its top is the channel. So let's view this. So how is this a channel protein? You'll see that some chaperoning complex. Or I don't know, but maybe it may not be a channel protein. I'm not sure. I mean, because it had these circular uh, empty space in between. I assume that it's a channel, but I actually don't know the details about the biology of this protein structure. But it was one of the protein structures that I had in my list. I have a test data set. This was one of those largest proteins, which contain tens of thousands of atoms in it. So if you look at this protein, its shape is really like very nice, regular, symmetric shape. But if you look at it, all these different colors that you see around, these are all different chains. It has many subunits. Okay, and if you look at from the center in from a certain orientation, you see that it has a like cylindrical hole in it. So as I said, I'm not sure that it's a channel protein, but it may be a channel protein. What happens is that channel proteins have similar shapes, and for example, and they act like they are situated in the lipid membrane. I think this is too big for the lipid membrane. If I look at these helices, maybe only one part of it, maybe this part, maybe in the lipid membrane, but as far as I know, the size of the lipid membrane, this many helices will not going to fit in that membrane. So this is really a large protein. Um, but we can maybe take a look at we can directly search for an ion channel protein. Ion channel. Yeah, it's a chaperone protein. That's correct. So thanks for the comment, Yammer. It's probably a protein that helps other proteins to fold correctly. Those are chaperone proteins. They are like helpers, and it's in its name it had the chaperone is probably a helper protein. You're right, Yammer. And it looked too big for a channel protein anyway. I mean, look at this one, which is much smaller than the other. So this is an outer membrane protein, open and closed formation. So if you look at this, yeah, I mean, this makes more sense. I mean, look, considering the previous one, this is a much smaller protein, but still it contains maybe hundreds of atoms. It's composed of all, so this is interesting. I know, for example, um, GPCRs are not, I know, for example, G protein coupled receptors, which are not like these ion channel proteins, but they are composed of helices. But if you look at this, it also has a really symmetrical shape, and it has like a cylindrical hole in the middle. So this is, I think, the ion that is transporting probably. What happens is that this is again situated. Imagine that this is situated like this in the lipid membrane. One part looking at the extracellular part, and the other is intracellular. Um, I don't know which part is which. Maybe it's this. It's like this. So I don't know which part is extracellular, which part is intracellular. But what happens is that if you look at this, if it's in the lipid membrane, this is like really a channel inside the membrane, like a canal, and ions pass through only chemicals, compounds with certain properties are going to be allowed to pass through here, again through really physical interaction between the protein itself. The protein maybe is going to, one of these is going to interact with that ion and going to capture it. It's like just like a grabber. It's going to grab it and put it then with certain, again, physical reactions, it's going to be put into the cytoplasm. So there are channel proteins like this. There are proteins that form the cytoskeletal structure. Many proteins act like enzymes. There are transport proteins, proteins that interact with the DNA to turn on and off certain genes, which we call those proteins transcription factors. So many functions that you can think of, very, very essential functions that you can think of in the protein, like for example, the ribosome is a protein, okay? Uh, so ribosome is a very also a large protein, which is responsible for translating the messenger RNA into a protein. So in protein synthesis, ribosome, which is also a really big macromolecular structure, is also very important. So everything you can think of is it's the I mean maybe from before this course when you heard the name protein, it was just something with some nutrition value. Okay, you need to eat proteins. So while we're getting proteins, actually, although we don't use those proteins as they are in when we eat them, you're just using those amino acids basically as building blocks to make our own proteins. Each body, I mean, a species, an organism has to make its own protein. I'm not sure whether a protein that you can get outside can be directly used. Probably it can never be used. But there are always exceptions. Biology is, whenever you make a statement in biology, somebody make up, oh, okay, this organism does that. This organism, so there may always be exceptions. So be prepared for that. But other than that, as I said, the protein, we have proteins that we eat. We use them for their amino acids, mostly their building blocks. We need if you want to make up our own protein in our body, and if they're so important to those cells' functions, we need to be able to synthesize. Your body synthesizes all these proteins at the moment at very fast speed. So that's the factory is working, and it needs resources. So those proteins are like those that you eat, provide the resources, the raw material amino acid for your body. I have talked about the levels of protein structure, but let's quickly go. So why do we study protein structure? Because as you can see, they may have many interesting, unique shapes. For example, I've just showed you one which is like cylindrical. Let me show you another protein. So here, uh, for example, let me show you a different function, a transcription factor. So yeah, look at this one, GCC box domain. So this one, transcription factors, usually they are they also have different addresses in the cell. They live in different components inside the cell. Some of them are in cytoplasm. Some of them, as you've seen, in the membrane. Some of them are in the nucleus. But if you look at this one, for example, it has it is really a different shape than that circular one we have seen. Look at this. So it has this shape, and this shape is probably also crucial. And so what we are seeing again is the cartoon view. We see the secondary structure. These local shapes are the secondary structure, and we see here what the entire protein. When you're viewing the entire protein like this, you're viewing the quaternary structure. Quaternary structure, it has probably I can see here two disconnected chains. One, yeah, I think it's a dimer. It's composed of two chains. Maybe it's a homodimer, which means that this chain and this chain is identical. If it's an identical chain of amino acids, two amino acid chains interacting to form, and they interact with each other in a unique way to form this shape, which we see here is the quaternary structure, and it probably interacts with the DNA like this in a certain orientation, with like two feet. It grabs onto the DNA maybe to do certain things. So I don't know. So for example, each of these shapes, let me show you another thing. Zinc finger DNA binding domain. So we're going to see a single, we're just looking at the domain here this time. It's very short, as you can see. This is like a finger of a protein. Some proteins contain this as they are, even on the chain, there may be certain domains, okay, certain regions in the sequence that turn into a certain shape. which has a very specific function. We call such places domains. So this is the zinc finger domain. So it is a loop like this and a short helix, and this may be the needed for that protein structure to interact with the DNA. Okay, so just to show you that there are really different uh, types of shapes, and this shape, the three-dimensional structure, is really crucial to its function. And most of the time, one thing that is really interesting in this is that you may see similar shapes, but if you look at their sequences, they may be very different. So, the structural shapes are more conserved in nature than sequence. You may find certain similarities that you cannot find in sequence. For example, you may take a look at two sequences and look at them and see that they have only 10% sequence identity. There are 90% of the sequences really different. And when looking at that, you may not consider them homologs. But if you look at their folded shape, you may see that their structure may be really similar to each other. And when you see the structure, then you may understand that, oh, actually, they are homologs. Okay, so structure is more conserved than a sequence, meaning that multiple different sequences may turn out to come up with the same structure. It's more closely, structure is more closely related to the function. It's not the geometric structure is not the only thing. So since the physiochemical properties of those amino acids are also important, which means that sequence also plays some importance here. It's not just a three-dimensional shape, but three-dimensional shape is determined by the sequence. So sequence is important, but certain differences in sequence which you may not observe when you look at the sequence only may be apparent when you study structure. So this is what it's saying. So this one, as you can see, this slide is old. This April 10 was probably like 4 or 5 years ago. There were like 42,000 structures. Now it's like, I remember it's like 170,000. It was something like that, right? So it almost increased by fourfold. Let's see. Yeah, it's 177,000. So yeah, it's increased fourfold since in the last 4 years, for example. So it's really increasing rapidly. So I was talking about just X-ray crystallography and nucleic magnetic resonance as the experimental methods to determine protein structures, but we have now new methods like cryo-EM. Cryo electron microscopy is also a really promising technique, which promises to find the protein structures in their native state. When you crystallize them, for example, you may actually break their natural habitat, and they may, in crystallized form, they may not be in their native state, which is not going to be very different from the other shape. But as I told you before, they may make small conformational changes or vibrations, and crystallizing them may disrupt those things. And in cryo-EM, cryo-electron microscopy, for example, you can try to view them in their native environment. So it's growing. So this slide is from 2000. So it's protein structures. We know more and more protein structures are solved experimentally every year. I've talked about the basics of protein structure, but I haven't talked about this chemistry. But we have seen when we looked at the PDB file example last week, you've seen these three atoms that were really crucial, this N, carbon alpha, and this other carbon. So this central carbon, and this forms, oh, by the way, these atoms, these two carbons and this oxygen, they are common in all of the amino acids. So that's a really interesting thing. Every amino acid has these four atoms as a common backbone. What makes an amino acid different than the other amino acid is this R part. So therefore, sometimes amino acids are called residues. So this residue part, like the additional part, actually determines the type of the amino acid. the simplest case, this R can be just a simple hydrogen. Okay, it may be just a hydrogen here, which will give you the smallest amino acids. Was it glutamine? It's something that starts with G. So I'm I don't know which amino acid is which, as you can see. I'm talking about protein structures, but I mean, if you're a biologist, that will be a very bad thing on my side to not know what that protein and what that amino acid is, because it also determines its size. If you're going to build those protein structures manually by hand, certain amino acids, since they're smaller, they can fit those small locations much easier than the others. So the smallest this R can be is just a hydrogen atom, and the largest it can be contains maybe tens of other atoms. But this backbone is in every amino acid, and this commonality is actually what makes the bonding of amino acids with each other. So they have different parts, but like every Lego part has this circular thing that you can stick to the other. It's like this backbone is like that. So this is the part where you can connect another amino acid. Look at these two amino acids. They all have different residue parts, but this N C C N C C, it just goes like this. So this carbon atom makes a peptide bond with the nitrogen atom of the other amino acid. This is how they connect with each other. This carbon connects to this beginning nitrogen of the other. So it is an order. The chain, if you look at this chain, if you're just going to connect another N here, NCC, NCC here, if you look at this, the leftmost N is going to be empty, waiting for a C to bond there. Okay, so this is therefore called the N terminus of the protein chain, and it's not, there's nothing that binds there. So, but it will be ready to accept the hydrogen, the peptide bond here. But so this is the N terminus. This is the C terminus. So from the first N to the last C, there's the chain of amino acids, one after another, bound together like this. And these, although the lengths of these bonds are fixed, they are rotatable. So what makes this shape, for example, so when it's 0 degrees, it's called cis, and under 80 degrees is trans. And this is the angle that this bond makes. Here we can measure. So how do we measure that? For example, this three atoms here, they define a plane. And this other three atoms, they also define a plane. So the difference, the angle between these two planes may be your omega atom, okay, omega angle. And so bond angles, these are really important to understand its shape. So there are dihedral torsion angles, but these are really going into too much detail into chemistry. Just know that these bonds are rotatable. That gives you the flexibility of for amino acid chains to get into very interesting different shapes. Uh, they their variability in this bond length, the bond angle. The bond lengths are usually, it varies between, but for between carbon and hydrogen, it's always 1 angstrom. Between two carbons, it's 1.5 angstroms. And bond length is just a function of the position of two atoms. Okay, so it's, as you can see, fairly fixed. But the bond angles, they range, the dihedral angles, for example, they range between 100 and 180. It's a position of function of three atoms, and there are some phi, psi, omega, chi angles. So these are their Greek names. The plot that plots the phi angle with respect to psi angle. So there are famous plots, the Ramachandran plot of a protein structure. So where is it? Yeah, the Ramachandran plot is the plot between phi and psi angles, meaning that for each atom, so by the way, what is the phi angle? As you can see, the phi is the angle between this C and this C that they make, two bonds that they make. From N to N, it's called, from this N to this N, it's the psi angle. So they are different. So by picking on two adjacent amino acids, you can compute the phi and psi values of those two adjacent amino acids easily. And if you plot for every adjacent amino acid, it will be a point in this plot. If you compute this phi value, compute this psi value, it will be a dot in this plot. This is a different way of visualizing those dots. If there are many dots here, it will tell you that there's a region or there are multiple regions in this protein structure in which the phi angle is between -6 and minus 180, and the psi angle is like between 60 and 180. Okay, plus this. So this, looking at this, some structural biologists may understand certain function, certain shape features of protein structures, but just know them as these things as side note. I'm not going to ask what are the angles, name the angles that form the Ramachandran plot. No, I'm not going to do that, but just know that the bond is rotatable, and that gives this protein the flexibility and its shape. I talked about levels of protein structures, and there's a question by Ahmed. Let me read that. Can we represent every protein structure as a one chain of amino acids, or there are like branches in the sequence? So actually, a protein structure will be most of the time multiple chains of amino acids. So there's a single chain of amino acid that is going to form. We call that a protein chain or amino acid chain. And sometimes, most of the time, multiple chains interact with each other. Again, physically, they form this quaternary structure. So we are a single amino acid chain. It's called tertiary structure. Okay, so we are going to see that. And in that cartoony version you have seen, for example, this is a single small part of a protein, but when we have looked at the largest protein, many proteins are composed by multiple chains. So these are also very small proteins. But if you, in this one, for example, we have seen two chains. One of them is here, the other is here. So many proteins composed of multiple protein chains. And if you look at this, for example, one part will be the N terminus, the other is going to be the C terminus. It has two ends. It's really like a ribbon that has a start position. Look at, this is the start and this is the end, and look at how close they are in 3D space. The start, maybe if you just unfold it, it's going to be a long string, but that string folds onto itself like a cord, and in 3D space, as you can see, parts that are very far away from each other on the sequence, for example, this amino acid and this amino acid, they are far from each other. It's in one end, the other is at the beginning, for example, but they are close to each other in 3D space. So when it folds, these interactions, we see them. Okay, so, but yeah, protein structures, this is a single chain of a peptide chain of amino acids. It falls into a certain shape, and proteins may be composed of multiple chains like that. So the branches that is this level actually. The branches that you talk about, that you ask, is the quaternary structure is the top of the branch, which contains all multiple chains. The tertiary structure, when we talk about, we go down one level in the chain and we just look at a single amino acid chain. A single amino acid chain, its three-dimensional shape, is the tertiary structure. And if you look at the single chain and we identify parts of it, for example, you look at this, I mean, you don't need to, when I say, for example, the first three amino acids are in a coil, and starting from fourth amino acid, for example, to 20th amino acid, they are in helices. If I show you the structure like that, it's also a two-dimensional structure actually. Let me display file sequence. Yeah, let me go then, or the structure summary. I think it will be in the structure summary. Yeah, look at this. Yeah, these are, this is an alignment, not here, the secondary structure. Secondary structure may be in the sequence part, or maybe in annotations. No, annotations are functional annotations. When you look at the sequence, yeah, look at this. When you look at the sequence information, this is our PDB amino acid sequence. And when you look at, it says secondary structure here. Secondary structure is also represented as a single line. It's one-dimensional information, actually. It's just saying that these amino acids are part of a helix. These amino acids are part of a helix. Anything between those two are not unclassified. They are like sometimes called turns, coils, oh, sometimes turns, coils. They are really the connecting parts. But you can see the helices. But what I want to show you here is that secondary structure itself, its information content, as you can see, it's one-dimensional as the sequence. And this is the amino acid sequence itself is sometimes called the primary structure. So that's, if you hear something like that, what is the primary structure of a protein? That may be misleading. It has the structure keyword in it, but when we talk about primary structure, we talk about this amino acid sequence. So the sequence is the primary structure of a protein. So it's simply the amino acid sequence. Secondary structure is helix, beta strands. Those are the most common. I mean, an alpha helix and beta strands are the most abundant. I don't know. There may be very few different versions of these, but when you talk about the structural universe, you always talk about helices, beta strands. For example, there is a classification, categorization of proteins structure universe, and at the top level, based on shape, this is mostly based on their shapes. Um, and by the way, the name of that classification is scope. Let me write in the chat box. Structural classification of proteins. So when you go to scope database, scope protein, so structural classification of proteins. When at the top level, so this is also a hierarchical classification. At the top level, as you can see, all alpha proteins, all beta proteins. What do you think this alpha stands for? Alpha helix. So alpha proteins, it means it's all helix. The proteins that contain all helices in them. Proteins that contain all beta strands in them. So all beta proteins mean that they are composed. The ion channels that we looked at, which was this nice small cylinder, it was all beta, for example. They're all beta proteins, alpha and beta proteins, and another alpha and beta proteins. But this one is A slash B, A plus B. So what is the difference between those? We are going to see. The difference is that the beta strands, they can form the beta sheet. So a beta strand cannot be a sheet like this. If you view a beta strand like this, it cannot appear by itself. Okay, you have to have at least another beta strand so that they form these hydrogen bonds in between them to have this stable sheet-like structure. And most of the time, you have a bunch of them arranged in a sheet like this. Now, if I tell you, for example, if I ask you, this is the sequence, amino acid sequence is going to begin here, it's going to end here. So this is the N terminus, this is the C terminus. Can we know, for example, whether the sequence, the amino acid sequence order is this direction? Or, for example, I can have something like this. Look at this. So it may be, the sequence may come here. There's a connector here and another connector here, and it may end like this. Okay, so the protein may look something like that. The amino acid sequence. And if you look at the order of these beta strands, they're order is anti-parallel. The adjacent ones here, if one of them anti-parallel in terms of sequence order. Okay, uh, this one in sequence, as you can see, I just follow the sequence order in a uniform from N terminus to C terminus. If you follow your sequence, you'll see that these beta strands are looking in, they're parallel, but they're looking at different directions in terms of sequence. For example, this will tell you that the first amino acid in this beta strand here, which is interacting with the last amino acid and the other beta strand. So they're related like this. So such kind of beta sheet formations is called anti-parallel beta sheets. Anti-parallel beta sheets. And the scope classification, this A plus B, means that it's composed of alpha and beta. So there may be helices here, another helix here. So if it's composed of helices and beta strands like this, and if the beta strands are like anti-parallel like this, its scope main class will be A plus B. So this plus is like there, and they are not direct, they're anti-parallel. They're, in terms of geometry, they are parallel, but if the order is different, it's called anti-parallel. So could we have, let me undo up to the point where I had only the beta strands without the sequence order. Now, I have the same beta strands, okay? Can they be facing all the same direction? Is it possible? Can I have the same beta strands and they are all facing the same direction, which we call parallel beta strands. So they are all, the sequence order is uniform in all three of them. This will be parallel beta strand. And in scope class, the scope class A slash B forward slash B is referring to this such kind of beta formation. So A alpha and beta proteins, A slash B is this one, and A plus B is the anti-parallel one. So as you can see, the entire top level class of the structural classification of proteins is done by the helices and beta strands that it contains. So these are really important local shapes. All the proteins they are your basic Lego parts, and how you connect them to find your different shapes is called the protein fold, a protein shape. All right. So by the way, I haven't answered this yet. So can we have such? You probably can draw a thing like that. For example, let me see. Now, what if I do this, and here I come like this, and then do that. You see, I was able to do that. So because we're in 3D space, they can cross each other. We're not trying to do a planar graph. We don't. So all these things, as you can see, these the turns, the coils that I draw, there may be a helix here. They are arranged three-dimensionally. So we have all the 3D space around them to have it come here, then a long part going this way, and again going this, and again the long, another helix, again this. There may be a helix here or not. Okay, there may be a helix here, and I may still, if you look at the sequence again now, starting here, following the sequence order. By the way, I have to increase my mouse pointer size. I forget that. Okay, so if you look at this, by the way, the mouse pointer size doesn't change in GIMP. Just noticed that, but the sequence order is fixed. Okay, I mean same. So this is called parallel beta strands. So this is how we form those beta parallel and anti-parallel. So you see here an anti-parallel beta sheet formed by two beta strands. Each of these things are called strands, and when they form this beta sheet, a beta sheet is a stacking of multiple beta strands. In alpha helices, every 8 amino acids make a hydrogen bond with the I plus fourth amino acid. So the helical shape, its stability, it stays as a helix. In order to stay as a helix, these hydrogen bonds, they help. And there's a turn. The helical turn is like every 3.6 residues. And as you can see, beta sheet, it also maintains its local structural stability, again with hydrogen bonds between these beta strands. And since the backbone is almost fully extended, meaning that it looks almost completely straight, but there may be small conformational, small turning. I mean, we say the beta sheet stays straight like this, but if you remember that ion channel protein, the beta strands were like turning around to form this channel. Yeah, in anti-parallel sheet, the sequence order is reversed. In parallel beta sheet, the sequence order follows the same in both beta strands. So here's an example of an anti. Oh, actually, it's also nice. In some beta sheet, you may observe multiple things simultaneously. For example, if these two are parallel to each other, actually, this one is also parallel. Look at these three. They are parallel, but if you look at these three, they are anti-parallel. So this is wow, very nice artistic rendering. I mean, compared to the ones that we have seen in Protein Data Bank, this looks really like more higher quality, right? It has some texture on it. And even shadows. We have shadows. It's probably ray traced. But it doesn't really look like this, the real protein. So it's just this higher quality rendering is, in terms of the biological information it gives, it doesn't give anything. It just looks cooler than the other one, okay? So this is, as you can see, there are 3 beta strands here. They all go in the same direction in sequence. And this is a parallel beta sheet. And this is an anti-parallel beta sheet here, where the beta strands are in different opposite directions. The other connecting parts between alpha helices and beta strands are called loops, turns. There's a distinction for sometimes they're loops, turns, sometimes coils. Turns are sometimes called coils, but everything is classified into the other things. So we don't have a different loop. We just name all of them turn here. So, and this table is an interesting table that shows the propensity, the preference of amino acids to be in a particular type of secondary structure. This doesn't mean that the alanine, for example, always has to be in alpha helix. No, they sometimes may be in beta sheets or turns. But if you look at this, their tendency to be in alpha helices is more compared to beta sheets and turns. Okay, so such a table, the preference table for amino acids to be in what type of a secondary structure. These tables, for example, there's a secondary structure prediction is also one of the first steps in protein structure prediction, which means that if I give you a protein sequence, can you solve this one-dimensional problem, which is can you assign certain intervals of this protein sequence and indicate whether they're alpha helices, whether they're beta strands, or whether they are turns. So this is called the secondary structure prediction problem, and it's very accurate for proteins which contain mostly helices. The difficulty comes, secondary structure prediction is difficult when a protein contains mostly beta strands, because beta strands, the difference you can actually immediately see here is that here in a beta sheet, the interaction between 2 beta strands to form a beta sheet, these 2 beta strands can be very far away in the structure, in the sequence. For example, this one and this one, in sequence, there's a big gap in between them, between this one and this one. In sequence, there's a big gap in between them, so they're not neighbors. In order to determine whether a certain part is going to be a beta strand, you need to look at the entire structure, which is more difficult than just looking at alpha helices, because if you look at alpha helices, the helix formation is really local. The helix, the hydrogen bonds that form the helix is between I and I plus 4, I plus 8. So it goes like this. So it's really a local structure and more easier to find. For example, a very simple secondary structure prediction algorithm, which uses these preference statistics, is this. You have a fixed window of, for example, size 15. In this fixed window of size 15, imagine you add up all of the preference values. And if I mean, you basically, it's like a majority voting. You ask all of the amino acids in that window. Do you want to be in a helix? Do you want to be in beta strand? Do you want to be in alpha helix? Do you want to be in a turn? If you ask the votes of those 15 amino acids in that window, and if the majority says, for example, we all want to be in an alpha helix, classifying that region as an alpha helix is not a very bad idea. So some actually the secondary structure prediction algorithms, they use this table to predict the secondary structure. So I will quickly give examples of tertiary structure and have a break after that. So the three-dimensional shape of an amino acid chain is the tertiary structure. So here's an example. Tertiary structure, again, the helices in this representation, they're shown as cylinders. And quaternary structure is the actual functioning protein in which multiple protein chains interact with each other in a certain way. So this is also important. For example, two proteins, are they going to be bond together from this end, or are they going to be bonding on top of each other in a different way? For example, look at these four. These four different chains, they are arranged together like in this fashion to form the functioning protein structure. But you can have this usually when we have these discuss about these quaternary structures, we call them, for example, we call this a dimer. Dimer means two chains interact together to form the functional unit. This is a tetramer, again using this Latin numbering system to indicate this. Tetra means four, so it's protein structure composed of four chains. But look at this. One way it could be like this. In another protein structure, it is formed, the quaternary structure is formed in a different way. So it's functional, how it's going to be functional. So quaternary structure contains also three-dimensional information, and additional three-dimensional information than just the shape of those individual chains. So this is the protein structure of hemoglobin, where we have two alpha chains. Two of them are identical amino acid chains. The two other beta chains, they're also identical. Okay. So yeah, that's it. We have talked about protein files. We have looked inside the PDB file earlier, and we have seen information about the atom and the amino acid as in these columns. The XYZ coordinate of that atom, nitrogen atom, is this. So these XYZ coordinates, the 0, 0, what is the origin here? Are they with respect to the Greenwich coordinate system? Are those latitude, longitude? No. It's actually there is no universal central 0, 0, 0 atom that people use. This XYZ coordinates are really like local, in its local coordinate system. We just decide on a certain part to be 0, 0, 0. We define our XYZ axes locally. So in its local coordinate system, these are the XYZ coordinates are these. So their actual values, therefore, is not really that important, right? What if, for example, I want to move this protein 20 units to the right? You add 20 to all the X coordinates. If I make this 30.8, 32.0, 32.06, 11.15, not 31.15, 32 point. If you just add 20 to the x coordinates of these values, is it going to change the structure of the protein? No, this is just a translation. It will just move your protein 20 units to the right. Its shape is going to stay intact. So therefore, these coordinates, their actual values are not that important, but their values with respect to each other are important. So we are going to talk about those. Let's have a 10-minute break, and I'm going to continue with the protein structure prediction problem after the break. I will stop recording now.